team of Zurich. And we are going to tell you how we taught ourselves how to add numbers. But first, I'd like to shortly introduce you who we are and where we come from. I'm sure you all know that Europe is here. And we will find Switzerland in the heart of Europe. So let's zoom in and find out something about Switzerland. Yeah, sure, there's not only cheese, but also mountains and chocolate in Switzerland. Yeah, now that you know something about Switzerland, we will come to the team. This is our team. We are 5 undergraduates and 4 graduates from very different disciplines like, of course, biology, and also engineering disciplines like computer science, electrical engineering, and biomedical engineering. Now, these are the contents of our presentation now. It has 2 main parts, each of them containing 2 sections. The first 2 sections are about the engineering point of view and contain how we found our system, how we decided what it should do, how we designed it and modeled it, and then about the simulation. Then the third and fourth part are about the biological implementation of the system and how we tested it and where we stand now and what is still left to do. Arthur is now going to hold the first and the second part.
Thanks, Franz. Hello, everybody. I want to start by introducing the direct application of our system as we built it now. The idea is to have a plate which is covered with mutated E. coli and expose this plate to patterns in 2 different ways. In our case, it's by light, projecting this pattern onto the plate and drawing it on the plate with IPTG, a chemical, and the cells are supposed to turn green where both of the stimuli have been present, and red where exactly one of the 2 stimuli has been present. Now, in a bit involved fashion, this is related to adding numbers. You can add numbers mentally, of course, or using a calculator, or you can do it on paper. And this is very, here you can see a bit how we add numbers, or could add numbers with our system as well. This is in decimal now. You start adding the numbers on the right-hand side, position by position, writing down the rightmost digit of the sum of in each position as a part of the result, and carrying over the overflow to the addition at the next position. Here we got 2 at the second position, so the result is 22. Now the same works for binary as well. This is really exactly the same. It's just another base, a bit uncommon for some people here maybe. There's nothing to be scared of. The result would be 4 in this example, 1, 0, 0. To see clearly what logic connects the part, these are the 2 operands for the addition at one position. Here both are 0, and the results, the one that's carried over to the next position and the one that goes into the final result. And I collect them now for all combinations that can occur, and we see for when both operands are 0, it's 0, obviously, the result as well. And if either of the 2 operands is a 1, then the sum is a 1. So we get here a 1 to the final result and a 0 that's carried over. And in case that both operands are 1, the sum of them is 2. So we get 1, 0, 0 to the end result, and 1 carried over. Now looking at the logic that can be used to derive these or obtain these 2 bits from the operands, we see that this logic is equivalent to so-called exclusive OR, which is gives a 1 when either one of the inputs is a 1, but not if both of them are a 1 in particular. And the AND gate can be used to calculate this carry bit. An AND gate means simply that the result is a 1 when both operands are 1. Now, out of these two logic gates, so-called half-adder can be built, and it has two inputs, the two operands, and the two outputs, the part for the end result and the bit that's carried over to the next position. And actually, complete addition units for binary numbers can be built out of this kind of device. And this is what we wanted to come up with in our biological system. Now, to get a complete system which doesn't only have pops in and outputs, we added parts from the registry, namely a light sensing device that was built by the Colliroid project, also an iGEM project, and the IPTG chemical sensing device, both from the registry and common fluorescent protein reporter devices. So yeah, they were connected to the inputs of our logic device, and the outputs were connected to the fluorescent protein producing devices. To come back to our pattern matching application, we see now here a pattern of IPTG in lines. I can't track them really. And another pattern of light, which is orthogonal to the first one. And now in the result we want to see in the end is that the cells produce green fluorescent protein where they were exposed to both stimuli, as I said before, and red fluorescent protein where they were exposed to either of the two, so either IPTG or light. To design these gates, we used an approach where we started out with concepts which were only concerned with the connection of the biological devices, but not with the specific biological parts used here, or building blocks. These are only two. We had 3 or 4 concepts initially. But we didn't have the resource to try every one of them. So we had to decide early on, only based on these concepts, and which one would be worth implementing. And the requirements that must be met by the system, and based on which we could make this decision which one of these gates we should implement, are, first of all, it should be biologically feasible. If we can't implement it in our bacteria, there's no point in following that approach. Then we need an appropriate steady-state behavior, so it actually behaves like the logic gate that we want to implement. Then it should be very robust to uncertainty, because, as was pointed out in former presentations, there might be quite some difference in the actual behavior of the biological system and the simulations we conduct. So by designing for large or high robustity margins, our chances are better that the design will actually work in the implementation. And it should also be appealing. Now, these two criteria can be evaluated or estimated using mathematical modeling of the device. And we did that using state space models, where you have a function which gives you the rate of change of concentrations of all the species that are present in the system, or we are concerned with at least. Now, this function depends on various other things. First of all, stoichiometry and the reaction rates, which in turn depend on concentrations, inputs, and kinetic constants. Yeah, this is the kind of model we built. If you want to see the detailed models, you can go to our wiki. For this kind of models, there are rarely closed-form solutions, but you can integrate them over time using numerical methods for specific initial values, obviously. This is an example now of an arbitrary 3-state system. We see the initial values of the 3 states, and we start simulation at time 0. And for typical system, we would expect the concentrations to converge towards so-called steady state values, where they remain over undefined time. These steady states were what we were interested for our outputs, because they're very convenient to measure. We don't have to measure at a specific point in time. They are also very robust to disturbances. If you disturb the system, it will return to the steady state. And they also exist in virtually all practical systems. Now, here is what we get as a result from a number of simulations. The input, one input is shown here, the level of one input. The level of another input is shown here. And the steady state value of our output state of the system is shown in the vertical axis. So what we can see here, this is just a hypothetical system, is that this system would give a low output when both inputs are low, when both inputs are high, and a high output value if either of the inputs is high. So this is a good XOR gate, which we would like to achieve in our concepts as well. We simulated these concepts now and got these results for the two concepts. And we see here pretty clearly that this concept performs a lot more like an XOR gate than this concept. So yeah, this was already a strong indicator of which concept we should choose for our actual implementation. Another kind of analysis that we were able to do with these simulations was to manipulate the parameters. Since this was our only concepts and we didn't have specific biological components, these reaction rates and everything had to be guessed, and yeah, there could be anything. So, but for this concept, it was far easier to find a set of reaction rates and other parameters for which it would behave like an XOR gate, whereas for this concept, it was pretty far to at least obtain this kind of behavior. There is another kind of analysis that can be used to estimate robustity towards parameter changes. This is called steady state sensitivity analysis. What you do is you actually measure the influence of a parameter to the steady state value. And it can be algebraically derived from the modeling we already did. Now, to get this point a bit more clearly what it actually means, if you had a sensitivity of 2 between some parameter and some output state, this would mean that if we change that parameter by 1% from the value we assumed initially, the steady state would change by 2% from the value that it was at initially. So we get this kind of relation, relative parameter change times sensitivity gives the steady state change. Now here are the results for the XOR concepts formerly shown on the left-hand side. We see with different colors all the different parameters we had in our models. The leftmost two columns are not parameters, but they are input levels, because the sensitivity also depends on where you are, at which inputs you evaluate this. Then here's a placeholder, and so these are different parameters at different input configurations. And what we immediately can see here is that these two parameters have a sensitivity of almost 0 compared to the other parameters. So we probably don't have to worry about them at all. Yeah. Now Olga will tell you a bit more about the biological implementation of what we did.
This slide shows the overall XOR system we designed based on our modeling results and also based on biological feasibility considerations. And it contains 2 main DNA fragments, as you can see here, which produce transcription factors, here and here, and 2 proteases in the middle. Our system was inspired by bacterial 2 hybrid systems, which can generally be used to detect protein-protein interactions. So for example, when protein X and Y interact, That brings the DNA binding domain, which in this case is the lambda C1, and the transcription activation domain, which here is the N-terminus of the alpha subunit of RNA polymerase together, and that results into functional transcription factor, which can be used to activate transcription of a downstream reporter gene, for example. However, in our design, we've used the DNA binding domain and the transcription activation domain directly, using a linker, and in this case, it has a TEV protease cleavage site. So when the TEV protease is present, it cleaves at the site, and thus the transcription factor is no longer active and cannot activate transcription of a downstream reporter gene. In addition, we had another transcriptional factor, which had exactly the same DNA binding and transcription activation domains, but in this case, the linker contained the TVMV protease cleavage site. So in this case, the TVMV protease would cut at this cleavage site and thus disable the transcriptional factor. So coming back again to the overall system, when there are no input signals at the two parts here, then nothing is transcribed, so there is no output of the system as expected. When we have one input signal, that produces, in this case, the transcription factor with a TEV protease cleavage site and the non-corresponding TVMV protease. So thus the protease does not recognize the cleavage site, and the transcription factor is able to activate transcription of a downstream reporter gene, in this case, as you can see. Similarly, if we have just the second input signal coming here, we once again produce exactly the same transcriptional factor, but in this case it has the TVMV protease cleavage site, and we also produce the non-matching TEV protease. So once again, we are able to activate the transcription of a downstream reporter gene, and we have the output as expected. However, if we have two input signals, then the proteases recognize their corresponding cleavage sites, and they cut both transcriptional factors. So once again, we do not have any output of the system. So as expected, our EXO only has an output when there is only one or the other input signal and not both, as we would expect. And the advantages of our design are that we used two very specific proteases, so they have almost no off-target effects and cleave only where we want them to cleave. In addition, these proteases can be expressed in their active form in vivo without digesting our cells. And lastly, in the functional transcription factors, the two domains, the DNA binding and transcription activation domains, can be separated by different linkers and still have a functional transcription factor. So in this case, we could simplify our system by just using one promoter for our downstream reporter gene. Here you see the overview of our AND gate, and perhaps you would be thinking that the simplest way to implement an AND gate when you just want an output when two input signals are present would be using, once again, a bacterial two-hybrid system. However, we wanted to try something more original. So here you can see what we did. Once again, we have two main DNA fragments. And when we have just the first input signal coming in here, that results into transcription of the T7 gene. And we have a T7 polymerase. However, in this case, the polymerase is truncated and not functional because of an early stop codon that we have here. So we do not have a transcription of a downstream reporter. But when we have a second input coming here, that results into production of a suppressor tRNA. And this tRNA is able to recognize the early stop codon. And by incorporating a glutamine into the amino acid chain, it results into formation of a fully functional, full-length T7 polymerase. So then the polymerase is able to activate transcription of a downstream reporter, and we have an output. So as we wanted for our AND gate, there is an output only when both input signals are present. So as stimuli for our system, we used light and the chemical. And for chemical sensing, we just use a standard IPTG sensing system, so I won't go into the details of that. For light sensing system, we actually, in a true registry spirit, reused the system that was designed by the UCSF group last year, and we tested it. And here you can see the results. So in the case when the bacteria colonies were grown in light conditions, they remained light. And when they were grown in the absence of light, they actually became dark as was expected. So the light sensing system did work for us. And the next step would be to test our gates using two inducible promoters without seeing if the light sensing and chemical sensing works, just to see if our gates work themselves. And as a characterization, we would vary the strength and duration of inputs to characterize our logic behavior. And here you can see what we would expect for results. So on the x-axis is time, and on the y-axis is concentration of reporter protein. So for example, we choose RFP for our XOR gate and GFP for the AND gate. So when there are no input signals, you would expect the output of both systems to be relatively low. And upon presence of the first input signal, either light or chemical, we would expect to see the output of the XOR gate rise and the output of the AND gate to stay still low, because the XOR responds to only one input signal and AND should respond to two. When we introduce the second input signal, as I said, we would expect the output of the XOR to decrease and the output of the AND to rise. So this is something we would like to see in the future. So this is the status of our system right now. We have already designed and ordered DNA for our XOR and AND gates. We have tested the chemical and light sensing systems separately, and they have worked. And also we cloned already the reporter genes that we would use for the separate gates into their respective plasmids. Currently we are at the stage of assembling the gates because they were composed of several parts. So we are at this stage right now. And then, as I mentioned, we would like to test them separately by using the two inducible promoters without incorporating the light and chemical sensing systems yet. And then the last part would be to combine the entire system together with both the sensing parts and the gates and test it. So in summary, our group designed the half adder, which can be used to perform addition in bacteria, or more practically could be used for things like was mentioned in the beginning as pattern recognition. We also developed two gates, the XOR gate, which was implemented using two very similar transcription factors and two specific proteases, and the AND gate, which used the original idea of a suppressor tRNA. And lastly, we learned a lot and really enjoyed the process. So we would like to thank the organization of iGEM and the following people for helping us a lot. And if there are any questions, I would like to invite the rest of our team to come and answer them. I'm so nervous. Did you hear it? Yeah. Questions? Yeah. Can you say it again? Not yet, because we haven't actually assembled the, oh, sorry. The question was if we have an idea of a response time of our circuit, and we haven't assembled the entire system yet. One of the reasons I said we would like to test the XOR and the AND gate separately with just the inducible promoters is to see the response time of that. And then once we incorporate the chemical and line sensing, I think that would introduce delays, but no specific answer yet.
Actually, we have estimates from the modeling side. We expect like somewhat around 12 hours to reach steady state, actually.
Okay. Yes?
I'm not sure. It's sort of along the same lines with the proteases for the XOR gate.
Yeah.
About how fast you think the type of protease versus if they work at roughly the same rates,
Mm-hmm.
or if you think the rates, if they were different, might have an effect on if only one shuts off.
The rates are actually quite similar. There's only a rate difference of a factor of 2, which is at this speed not very crucial, because anyway the protease are much faster than the transcription factor can be produced.
Any more questions? Okay.
Somebody did a similar experiment using ribosomes.
I don't think so, no. Oh, sorry. The question was that there was similar work done with ribozymes and whether we have looked at that body of work to base our design on, but I don't think so, no. There in the back?
I think your XOR design is really nice because it's so symmetric between the two inputs.
Mm-hmm.
Do your models tell you what the downside is when having very asymmetric inputs in AND gate? Two input groups have different things.
I didn't quite understand.
Design is very asymmetric between the two inputs.
The AND gate.
Design is very symmetric between the two inputs.
Yes.
Do you know from your modeling whether having these very asymmetric inputs in the AND gate somehow compromises the design?
Okay.
That's actually a very good question. It was whether we expect asymmetric behavior for the AND gate because it shows asymmetric design. And we also thought that it would come up with a asymmetric behavior, but our modeling results are pretty symmetric. What we can say is that the sensitivity shows this asymmetry which you see in the models.
All right. Any more questions? Yeah. Okay. So the question was, in case we could choose for our system as the two elements to be sensed, what would we like to choose for that? Actually, I don't think we decided because we wanted to be, we wanted our system to be so flexible. And in the process of, actually initially when we were selecting a project, we had several ideas of something like the presence of some rare metal that we would like to get or something far off ideas, but nothing specific. Yeah. Okay. All right. Okay, thank you.